Hello everyone, so today we're looking at cell and tissue architecture. We'll be focusing on the composition of the cytoskeletons, the cytoskeleton and its motor proteins, and the extracellular matrix, it's also referred as ECM. So tissues and organs. So tissues and organs are communities of cells. So these are kind of like the levels of structure, but you'll see this in a lot of physiology as well. So a tissue is a collection of cells that work together to perform a specific function. Two or more tissues combined, functioning together, form an organ. So cells, tissues, and organs have distinct shapes. The form fits the function. So again, cytoskeletons and animals give us our cell shape, and the extracellular matrix and cellular junctions allow for structural integrity of tissues and organs and allow for adherence as well. So example is the skin. In plants, the extracellular matrix is important in cellular shape and adherence. So, the cytoskeleton. So the protein fibers of the cytoskeleton provide the internal support for the cell. So here we got different various pieces. We have our microtubules. So again, all eukaryotic cells have microtubules and microfilaments. So we've seen these in, you probably recognize these from muscles. Animal cells also have intermediate filaments, including protein subunits. So here's an example of our protein subunits. Here's an example of microtubules. So the centrosome, which we'll talk about in our DNA, cell division, and then here we have our microfilaments. So microtubules. So we can see a microtubule is formed of two components, an alpha tubulin and a beta tubulin, which forms a tubulin dimer. So we can kind of see this, the light blue and dark blue. So microtubules are polymers, which we talked about in our proteins, which is multiple monomers together of protein dimers. So each dimer is made up of two tubulin proteins, alpha tubulin and beta tubulin, which you can see here. So some examples of microtubules or in animal cells are the centrosome. So the centrosome helps maintain a cell shape and allows it to withstand compression. It's very important. Microtubules also provide tracks for the transport of material, and they're found in the spindle apparatus during cell division. They also make up cilia and flagellae. So if we see flagellae on bacteria, it's like the little tail. It's also located on sperm. We also have cilia, which we can see in our nasal mucosa. So these are the things that kind of help move different dirt and debris out of our airways. So for example, here we can see coordinated beating of the cilia that cover the paramecium moves the cell through its environment. That's one example. The cilia in these human airway epithelial cells, which I mentioned, propel mucus containing debris out of the lungs. These unicellular algae are propelled by two flagellae. In some cells of multicellular organism, including sperm cells, swim by the movement of flagellum. So the microfilaments. So microfilaments are polymers of actin monomers in a form of helix. We can see that kind of helix structure here. That's a very bad version of it. So these are the thinnest of our components of our cytoskeleton. They are relatively short and extensively branched just beneath the plasma membrane of a cell. So the microfilaments are important in reinforcement of the plasma membrane and help organize the membrane proteins. So here's our examples. So here we can see the circumferential band of microfilament bundles, and this is an intestinal epithelial cell. So here's a microvillus, which we have in their stomach, kind of like that. We'll see that in physiology when we get there. So here at plasma membrane, the cross-linking actin-binding proteins in the actin microfilaments. So I'll just zoom in for a second if you want to see. So again, filaments, and then our binding proteins. So microfilaments are found in the microvilli. So here's our microvillus, which are in the intestinal cells. These are longer bundles form bands around circumference of the epithelial cells. These microfilaments also take part in the transport of materials inside the cells, the shortening of muscle cells, like in contraction, which we'll see in physiology with our actin and myosin. They involved in the separation of daughter cells during animal division, which we'll also talk about. So So microtubules and microfilaments are dynamic. They're not just static, rigid bands, essentially. So these subunits are added and lost from the ends constantly. So we can see here, pieces are being added and removed constantly. See, these usually these polymers grow faster at one end than they do the other. So here we have the plus end is fast growing and the minus end is slow growing. Again, here we just said that. So in animal cells, the minus ends are at the organizing center of the centrosome. The plus ends project outward toward the plasma membrane. So the plasma membrane is likely exposed to more, activity. So that's why it grows faster, to help it deal with its more challenging environment. So dynamic instability. So microtubule plus ends, so those are the ones that grow faster, also undergo random cycles of rapid shrinkage. This is called depolymerization. This is followed by a slower growth called polymerization, which causes dynamic instability. This dynamic instability in the centrosome, for example, allows spindle microtubules to quickly find and attach to chromosomes during cell division. So this is important in our division to allow this to happen. So motor proteins, microtubules, microfilaments, and movement. So the movement of microfilaments and microtubules on their own by polymerization and depolymerization is limited. But when they are joined with motor proteins, they are able to cause large movements. Both microtubules and microfilaments can act as tracks for these motor proteins, and these motor proteins move substances in the cell. So essentially, here's a protein, and that usually you can see figures and videos of these. They kind of walk with like feet, and they'll move along the proteins to the other cells or other molecules. So here's a kind of example. You can see those little feet I was talking about. They move along. So one example is kinesin. So kinesiology, kin is movement. So we have a kinesin protein which carries cargo such as vesicles toward the plus end of microtubules. So here's a rapid growing end. So these kinesins will carry something such as vesicles toward the end, and you can see here using energy through ATP, they walk slowly along to the other side. So these motor proteins, like I said, kinesin and dynein. Kinesin, which moves the cargo towards the plus end, and dynein moves towards the minus end. So dynein minus, kinesin positive. So these motor proteins undergo conformational changes and are powered by ATP. I recommend you look up a video of these. It's quite interesting. So another example of kinesin and dynein at work. So in the color changes seen in fish and amphibian embryos, such as the zebrafish embryos. So this occurs with specialized skin cells called melanophores, which move the pigments. So these move the pigment granules in response to hormones or neuronal signals. So when these motor proteins move these pigments, again, kinesin and dynein. So when this movement occurs, so again, movement of the pigment along the microtubules with the help of kinesin and dynein. In this case, kinesin moves granules to the plus end, like we said, in the microtubule, and in this case causes dispersal, and this occurs at night. Dynein moves the granules to the minus end, causing aggregation during the day. If the embryo remains dark after daybreak, so essentially what we're saying is that at night, there's a hormone that knows it's at nighttime, causes kinesin to move towards the positive end, causes light to be emitted. Essentially, these colors and light. So here you can see. Then dynein moves the granules to the minus end, and we have the opposite. So here, in the dark, melanin, which is another protein, so similar to melatonin, melanin granules are dispersed outward by kinesin, causing the embryo to be darkly colored. Sorry, it's the opposite. So during night, it gets dark. Essentially, is this, and then in the light, melanin granules are aggregated toward the center by dynein, causing the embryo to be lightly colored. So it allows it to change color depending on its environment. So there's an example. We can see the transition between light and dark. So these, like I said, these microfilaments can also act as tracks. They can move cellular cargo, such as transport vesicles. Muscle contraction also uses microfilaments. We talked about actin and myosin. And these motor proteins, for example, myosin moves towards the plus end of the microfilaments. So here's myosin, and here's our actin filament. So we have our myosin heads, which essentially we can see them moving along this can cause muscle contraction. And again, ATP is needed to drive this. Another examples are microtubules in the cilia and flagellae. So microtubules in the cilia and flagella are arranged in a 9+2 arrangement. So here's that 9+2 arrangement. That's what it's referring to, the way they're, so we can see here, a cross-section through an individual cilium reveals the 9+2 arrangement. So it's got 1, 2, 3, 4, 5, 7, 8, 9, plus 2 in the middle. So that's where that comes from. There are 9 pairs of microtubules along the periphery and 2 microtubules at the center. We have these cross-linking proteins connect the outer doublets to the center pair, and then dynein molecules connect doublets to each other. Again, ATP powers the motion of cilia and flagella, causing that wave-like motion that we see. So we can see here an electron micrograph shows that cilia, paramecium with microtubules at their cores. And again, here's the anatomy. A few more. So again, we have our dynein arms, which are these little arms. We have cross-linking proteins, which you can see here. We have a central microtubules right there, and then our microtubule doublets throughout. And there's 9. Remember the 9+2 rule. So actin and tubulin are evolutionarily conserved. So actin and tubulin are found in all eukaryotic cells. Amino acid sequences of yeast tubulin and human tubulin are 75% identical. Similarly, amino acid sequences of amoeba actin and human actin are 80% identical, giving us insight in some of our evolutionary origins. So prokaryotics, for example, also have a system of proteins similar in structure and function to cytoskeleton of eukaryotic cells. Also, at least one of these cytoskeleton-like proteins is expressed in chloroplasts and mitochondria of some eukaryotic cells. And this supports the endosymbiotic theory. So now we're looking at the intermediate filaments. So these intermediate, they have an intermediate diameter. They are polymers of intermediate filament proteins that combine to form strong cable-like structures. And again, these are mainly for providing mechanical strength. So for example, the proteins making up intermediate filaments differ from one cell type to the other. There are many different kinds, hundreds of different actually. So again, an intermediate filament is a strong fiber composed of intermediate filament proteins. And then here we can see it contains a fiber subunit. So the analogy we have here is that steel cord, where it kind of works in the same way. Whoops, sorry. So one example is keratins and laminins. So we can see there's intermediate filaments composed of keratins in the cytoplasm of the stained red. Then we have the composed of laminins in the nucleus are stained blue. We also have association with desmosomes, which provide strong support for the cells. So here we have our skin, epidermis, basal lamina. Here's a melanocyte. Here's our keratinocytes, so this is in the skin. We have our intermediate filaments and our desmosome. So the strout-like intermediate filaments link the desmosomes and keratinocytes. Linked in keratinocytes provide a great deal of structural support to the skin. So these really help with our structural integrity, as we mentioned. So one example is an intermediate filament defect called Epidermolysis bullosa. So this involves a defect in intermediate filaments, which make the skin less resistant to physical stress. So individuals with this condition have defective keratin genes. As a result, intermediate filaments do not polymerize properly, weakening the connections between the layers of cells that make up the epidermis, and they can result in these unfortunate lesions. So summary, we have cytoskeleton elements here. We have our microtubules, which have tubulin dimer subunits. Their major functions are involved in cell shape and support, cell movement, example in cilia and flagella, cell division for chromosome segregation, which we talked about. We have vesicle support, and we have organelle arrangement. Then we have our microfilaments, which are made up of actin monomers, which help in cell shape and support, help with cell movement by crawling, which are those motor proteins. They're involved in cell division or cytokinesis, splitting of the daughter cells, vesicle transport, and muscle contraction, like we said, actin and myosin. We have our intermediate filaments, which are quite diverse, but they're mainly involved in cell shape and support, like that steel cable we saw. So again, extracellular matrix. So again, this is also referred to as ECM. You see here. So far our cytoskeleton is involved in cell shape and structure, and our cell junctions allow cells to stick. Example, tissues forming a tissue. So our extracellular matrix provides molecular framework for structure. So the extracellular matrix is an insoluble meshwork made up of proteins and polysaccharides. It is secreted or modified by many different types of cell, and there are many different types of ECMs. They differ in the amount, the type, organization of proteins and polysaccharides that make them up, and this contributes to the structural support. So, for example, in plants, the ECM is the cell wall, which is made of cellulose. This cellulose provides support by forming interconnected network between cells, and this composition is in the middle. It forms the middle lamella, the primary cell wall, and the secondary cell wall. So you've probably heard of cellulose in grass. So here's my bad grass drawing. And this is something that cows eat, and we can't eat. We cannot digest cellulose. So this is a lot of certain plants that have this we can't eat, but certain animals can eat it, such as cows. So again, we have our middle lamina, primary cell wall, and secondary wall, then that plasma membrane. So the middle lamina, which is the outermost layer, is a glue-like complex carbohydrate which allows cells to stick. Then that primary cell wall is a thin, flexible, formed mainly of cellulose and other molecules including pectin. Then finally we have our secondary cell wall, which is found in many but not all plants, which contains many cellulose and lignin. Lignin hardens the cell wall and allows waterproofing, and this allows some plants like trees to grow very large. Cellulose is part of the ECM assembled outside of the plasma membrane. Glucose monomers and enzymes formed for assembly are transported outside from inside the cell via microtubule. So in animals, the ECM of animals is like plants is a mixture of proteins and polysaccharides. Its composition is mainly large fibrous proteins, including collagen, elastin, and laminin, and this gives it its tensile strength. There's a gel-like polysaccharide matrix, which is negatively charged. So here we can see cells and connective tissue of animals reside in complex meshwork of extracellular fibers. So here we have our elastic fibers, collagen fibers. It's probably easier to see in the graphic, but here's our elastin fibers, very thin and have elastic abilities, mainly form of elastin. We have our collagen fibers, which you can see are thicker and allow for more rigid. We have our fibroblast cells, fibroblast nucleus, and our gel-like structure, which is kind of this filling in of the space in between. So collagen. Collagen is the most abundant protein in extracellular matrix of animals and the most abundant animal protein on the planet. 90% of collagen is formed from type 1 collagen. There are several different types. Example would be type 1 collagens in the dermis of the skin, which provides support. Collagen is composed of intertwined fibers and polypeptides wound in a triple helix. So here's that triple helix. So here we have collagen fiber, then it goes into our fibrils. Then we have our collagen molecules, which form that triple helix. Collagen molecules are bundled like steel wires in a cable. So one bundle forms a fibril, like I said, and then these fibrils are assembled into fibers. This gives it quite a very efficient way of designing it, allowing it to have lots of strength and work extremely well in its function. So cell shape, and gene expression. So the structure of the ECM can influence the shape. So, for example, fibroblasts grown on a two-dimensional matrix attach and become flattened. And compared, when fibroblasts are grown in a three-dimensional matrix, they are spindle-shaped and look like they do in vivo, so like they're formed in our body. Another example is that neurons maintained in the absence of laminin attach to the surface but do not take on the appearance of a nerve cell. In comparison, neurons cultured on a surface coated with laminin develop axon-like extensions of the plasma membrane. So here we have that little neuron structure, the dendrites, and then our axon. And again, the composition of the ECM can influence the cell shape. The structure and composition of the ECM can also influence gene expression. So, for example, we can grow mouse mammary epithelial cells in milk, inducing hormone prolactin. These cells express genes for milk proteins. We can grow the same cells on a plain glass coated with collagen. and these will grow and live but secrete only small amount of milk protein. If we grow the same cells in a 3D collagen gel, we synthesize and secrete 10 times more milk protein. So we can show how important the environment is, not just the cells themselves. So yeah, and again, this is a pretty good fundamental lecture for moving into physiology. We'll talk quite a bit about these proteins, especially when we move into some muscle stuff, and a lot of these structural proteins and motor proteins are quite important. So I hope you guys learned something. Take care.